Hello, everyone. This is Dr. Wiseman from Biology 104, Intro Physiology. Due to the weather conditions, I've elected to do this lecture online using video technology. I will have the slides available for you in Brightspace. A couple of notes about how we're going to proceed. First of all, you can see that I have a pointer that I can move that pointer around. So it would be to your benefit to take notes just as if this was a regular lecture, as if I was pointing to a certain area. Print out the slides, take quick notes. That way you don't have to watch the video all over again in order to get more information. The key is efficiency, right? The key is to do your best work the first time around so that your requirements to revisit the material like this are targeted rather than wholesale. So follow along with me. Good luck. And if you have any questions, please just let me know. I'll be glad to talk to you about the material. The first thing we're going to do is talk about some of the anatomy of the respiratory system. I do try to stay away from large amounts of anatomy, but in this case, it's important that we do cover some basic structures, talk about their functions as well. So the idea would be that you get to know a structure. It's not going to be super detailed, not as much as an anatomy course, but enough for us to ascribe function to it. And while we're working on anatomy, let's start with the whole system itself and get some functions for the entire respiratory system. We all know that the respiratory system is for gas exchange. That's something we've known probably since grade school. However, there are some other functions that you should be aware of and are incredibly important for patient well-being and proper function and occupation. So we have exchange of gases, most importantly, oxygen and carbon dioxide. Along the lines of carbon dioxide, part of what carbon dioxide's incredibly important role in the body, we think of carbon dioxide solely as waste. It's not. All right. It's very, very important in regulating body pH. Disturbances of breathing patterns and inability to move air not only affects blood oxygenation, but also affects body pH and body carbon dioxide levels, which again are as important to life as oxygen levels. So regulation of body pH. If you get out into the clinics, you might hear about the condition respiratory acidosis. Respiratory acidosis is where the breathing patterns are interrupted to a point where carbon dioxide builds up and pH goes down. You become more acidic. And you also have respiratory alkalosis. And the most common cause of alkalosis is hyperventilation. You're also protecting the body. Pathogens can and do enter the body through the respiratory airways and lung surfaces. That's because the lung surfaces are very, very thin. It means that there's not much distance to travel in order to get into the bloodstream. So we're going to have to protect from that. So from inhaled pathogens and irritating substances. These could be solid particles like smoke, or could be liquids, or could be dust, all sorts of things. So we need to protect these very, very fine, very, very delicate exchange epithelia from these pathogens and irritating substances. And the last one may not make, it might not be as intuitive as you might think, but vocalization, a very, very important physiologic role for maintaining physiology. You can say, help! You can't solve your own physiologic problem, so you need assistance from others, or in the case of reproductive function as well. So I like to say that psychology is actually the study of behavioral patterns in order to achieve physiologic and reproductive goals. So big four here: gas exchange, as we all know, regulation of pH, protection from pathogens and irritating things that might cause inflammation, and then lastly, vocalization. The process by which we move air, and we're going to break that down here in a little bit more throughout this presentation, is collectively known as bulk flow. That is to say, as opposed to say osmosis or diffusion, this is more of a macro process. What we're doing is we're varying pressures. It sounds kind of strange if you haven't thought about it. But what you do is you change the volume of your lungs. When you change the volume of your lungs, the pressure in your lungs changes. And when that pressure changes, the air moves in or it moves out in order to equilibrate with the atmosphere. So let's say we are exhaling. So we're reducing the volume of the lung. That causes an increase in pressure, however short, maybe for just a split second, and that increased pressure causes the air to move out of the respiratory system out into the atmosphere. So we're moving from areas of higher pressure to lower pressure. Without that gradient, you can't move the air. We do this with muscles attached to bones and connective tissue. Also, we have airways, and those airways have friction in them. So when you're moving through the airways, the air is bumping up against the walls of the airways, and that creates a little bit of friction that we term resistance. The smaller the airways, the more the resistance there is. And this is why in cases of pediatric respiratory disorders, it tends to be much more acute in pediatrics, not because the kids are more delicate or something. It's because of pure physics. Their airways are smaller. It's a physical thing. It's not a toughness thing. And so kids tend to be much more sensitive to changes in diameter of their airways. And that's a function of resistance, the ability to move the air in the first place. So now that we've gotten some of the general system-wide functions, we're going to just ever so slightly begin to narrow down a little bit. So let's start getting into some of these anatomical components. We have those airways. That includes the upper airways and lower airways. I have a slide for that coming up. These are meant to convey the air to the exchange surfaces, but also to condition the air in order to prevent pathologies or inflammation disruption of those surfaces. We have the exchange surface. This is known singularly as an alveolus, a single alveolus, and collectively the millions that we have alveoli. This creates a very, very large surface area which allows for lots of interface between the gases inside the alveoli and the bloodstream. Lastly, we have bones and muscles. They provide a source of stability to allow for volumetric changes in the lungs. If you don't have an insertion point, a stable point in order to exert force, then you can't create bulk flow. So we have the conducting system, the airways, we have the exchange system, and we have the muscle and propulsive system, if you will, for moving the air in and out of the lungs, what we collectively call ventilation. So conducting system and bones are for ventilation, alveoli is for exchange. So now we're getting into a little bit of anatomy, and remember, it would be a really, really good idea to get to know these terms, not only in terms of what they are, can you identify them visually, but what they are doing. That's the essence of physiology. You have something like the nasal cavity. What is it doing? It's slowing air down, it's moisturizing the air and trapping potential pathogens and irritants. So let's talk about this a little bit. First thing we have is what we call the upper respiratory system, and the line is right about at the base of your Adam's apple. If you feel down there, you might feel actually there's a little soft spot if you're lucky to find it. That spot right there, right at the base of the larynx, is the conventional borderline between upper airway, the upper respiratory system, and the lower respiratory system, which includes your trachea, your bronchi, bronchioles, and alveoli in lungs. So make sure that you know where these things are generally found and try a little bit just to ascribe a function, a short function to them. We're going to talk about the upper airway collectively, the lower airway a little collectively as well. Starting with the upper airway, we have the nasal cavity, the pharynx, and the larynx. The goal of the upper airway, as I mentioned previously, it's entering through these. It can either enter through the oral cavity or it can enter through the nasal. Air actually varies a little bit depending on whether it goes through the nose or through the mouth. The mouth is a little bit less able to condition air and trap pathogens as the nasal cavity is. But in times of increased metabolic need, you may just need to get that air in. And so the mouth and the oral cavity can provide a larger airway in times of increased metabolic need. So we have the pharynx, which is just the junction point between the nasal and mouth cavities. It's sort of a common, shall we call it a vestibule, if you will, for both entries into the ventilation respiratory system. Then you have a structure called the epiglottis, which is designed to protect the trachea from foreign material. When you swallow, what happens is that epiglottis folds over top of the trachea to allow for material to go into the esophagus rather than the trachea. Choking situations, aspiration of fluids often occurs due to either malfunction of the epiglottis or somehow material getting in there during the inspiratory process. So the epiglottis is open, and so it goes down into the foreign material ends up in the trachea. The larynx, laryngeal, this is where we have vocalization structures. All right, it's right there at the top of your Adam's apple and involves that area. Mostly for airway regulation, vocalization is what the larynx does. So collectively, these passages are slowing down the air a little bit. They're slowing down the overall flow, so it can be filtered, and that's why the nasal cavity is slower because it has more structures and it's doing more filtering and more conditioning. What I mean by conditioning is raising temperature if it's coming in from the atmosphere, or at least getting it to a body temperature. Maybe it's too warm and trying to cool it down. Adding moisture and or trapping irritants, particulate matter, pathogens. So now we're into the lower airway where we're going to continue to filter, where we're going to continue to moisturize, just like what we did in the upper airway, though there's less emphasis on slowing the air down because we have hopefully achieved that in the upper airway. So we pass through the epiglottis, through the larynx, and now we are into a cartilaginous structure known as the trachea. Again, we're going to continue to filter. We're going to continue to moisturize and adjust temperature. We call that conditioning. And then we get into the lungs itself, and then we get a bifurcation into the right and left lungs. That structure there, a bronchus in singular or bronchi in plural. As a little trivia for you, if someone aspirates a foreign body, an object of some kind of piece of food, or it could be a small object accidentally, more often it will settle in the right side lung, in the right lung, because that right main bronchus is actually a little more vertical, and we're usually upright. We're usually either sitting upright or standing. And so, assuming that position, the gravity would pull it to the right, and the left has a little more of a horizontal deviation. And so, foreign bodies are less likely to lodge in that lung due to that sort of horizontal deviation. Then we have bifurcations yet again. We get the secondaries and tertiaries, and you actually have up to 24 levels of bifurcation. again and again. It's very, very complex structure. It's really cool to look at it in fetal development. When we get down to the very, very small, we actually start to get more muscle in there, and that is what we call bronchioles. We can actually contract and relax those in order to either adjust where the air is going in the lungs or adjust resistance, etc. Again, these passages are structured to filter and condition, but not necessarily slow it down. Now, once you get to the bronchioles, that story gets a little muddier. But nevertheless, what the conducting airways are doing is getting that air to the exchange epithelia. We still want to condition, we still want to filter, but not necessarily slow it down as much as we had in the upper airway. And so let's talk a little bit about the act of conditioning. How does that work? When we say the term filter, in my mind's eye anyway, I think of something moving through a mesh or a structure of some kind, and it physically impedes the movement of some kind of an object like dust or or particles, et cetera, et cetera. In this case with the lung, when we say filter, what we mean is more analogous to, say, trapping with a piece of flypaper. The air moves across these surfaces, and the surfaces have moisture and materials that will cling to and hold on to particles and pathogens as that air moves through those airways. And so we have epithelial cells all throughout this system, just like our skin, only these epithelia do a couple of things. They secrete mucin. There are glands that will release mucin, which is a very, we all know a little bit of a sticky, viscous material. It can vary. It can be very, very fluid if there's a lot of water in it, or it can be very, very viscous, like what we're familiar with our noses. In any case, mucin and other proteins, we have surfactant. It's kind of more of a lipid-like material. And certainly we move water to the surface as well, because we're not only trapping and filtering, but we're also conditioning, moisturizing. Okay. And so we have these glands that will add material to the surface of the airways in order to trap any particulate or pathogenic matter on the way to the alveoli. In addition to these glands, we also have cilia. And those cilia are there to take that trapped material and move it up to the epiglottis. So when you clear your throat like that, and then you swallow, what you're doing is you've brought material up to the epiglottis, you've dislodged it from the surface, and then you swallow it down the esophagus, which is a great way to actually have immune surveillance. The majority of our immune system is actually found in our digestive tract. So by clearing your throat and then swallowing, you're actually providing potential pathogenic material to your immune system. And so the combination of glands and cilia allow us to condition and filter the air in terms of pulling out particulate matter, introducing moisture, so that the alveoli are protected. So once the air has moved past the bronchioles, and remember that bronchioles contain smooth muscle, and then we talk about bronchodilators and bronchoconstrictors. That's what we're talking about. The smooth muscle in there can change the resistance. When we get to later on in the lecture, we'll talk about resistance by virtue of, they can change the resistance by virtue of changing the diameter of the bronchioles. But once we're past that, once we're in the alveoli, this is where we begin to see actual diffusional gas exchange. So they're very, very thin, all right, because diffusion depends on distance. The longer the distance, the slower the diffusion. And since we are highly metabolic organisms, we can't afford to have slow diffusion, if you know what I mean, when it comes to gas exchange. So these bronchioles, they look like little grape clusters, have lots and lots of surface area and are very, very, very thin. They're very, very thin, in order to allow maximal rate of diffusion, whether it's oxygen into the extracellular fluid or carbon dioxide out of the extracellular fluid. Notice how I said that, because there's actually a gap between the alveoli and the blood plasma. And so, again, keeping it thin, keeping that distance short maximizes diffusional rate. So as we're moving air in and out of the lungs, that process, sometimes we refer to it as respiration. Actually, that's not, in physiological context, that's not actually correct. When we say respiration in the physiologic context, we mean making ATP using oxygen. Here we are moving air using a bulk flow process. That is collectively known as ventilation. It's better than respirations. I know that people do conflate those terms, but for our purposes, let's keep respiration as a metabolic process of using oxygen, and ventilation is the physical process of moving air. So inhalation, what we're going to do is we're going to increase the volume of the lungs. That increase then causes a pressure drop, and the atmosphere equilibrates to equilibrate that pressure drop. Exhalation, what we do is we reduce the volume of the lungs. That causes a transient increase in pressure, and the air moves out, kind of like a tidal pool on the ocean. So the goal is to get that air in there to facilitate, and the way you would describe it is that we're trying to maintain a gradient here. We're trying to keep the oxygen content in the alveolus higher than the blood so that the oxygen will move into the blood. And we're trying to keep the CO2 lower than the blood so that the carbon dioxide moves out. A kind of an interesting way to describe gas exchange is to say it is the tale of two cities. What do I mean by that? Well, it's actually the tale of two surfaces. First of all, you have the lungs. All right, so you have a gas exchange between the alveoli and the capillaries. That's one city, if you will. This is where oxygen is diffusing from the alveolus, assuming proper ventilation and circulation, from the alveolus into the blood plasma and eventually onto the red blood cells, and vice versa, because CO2 is lower concentration here in the alveolus, that we have diffusion away from the blood plasma, away from the red blood cells into the alveolus. And so that's the story of one city, the city of the alveolus, if you will. And then the other city, the other location is in the tissues, where it's the opposite, where what we have is a low concentration of oxygen in the cells because the cells are metabolizing the oxygen. So oxygen concentration is always dropping in the cells due to metabolism, and that leads to a diffusional event coming from the red blood cells in the blood plasma, then the extracellular fluid, and eventually into the cells. And vice versa, CO2 levels are rising, which means that we are going to have a gradient towards the blood and a diffusional motion. So it's the tale of two cities. To this point, we've been talking about structures and movement. Let's talk a little more about pressures. There is a very, very important aspect that we need to discuss, and is quite common in emergency and care and acute care, and that has to do with the fact that and let me back up for just a second and say, in order to move air, we have to change the volume of the lungs. So we change the volume of the pulmonary cavity. That change then causes a pressure change with the air that's inside the lungs. Even if you're at your deepest exhalation, there's still air in there. We call it dead space. That air changes pressure, and then the atmosphere equilibrates. So if we're inhaling, what happens is we lower the pressure inside the lungs by virtue of volume, and then the air moves in order to equilibrate from the atmosphere. If we're exhaling, we are reducing the pulmonary cavity size. That increases the pressure of the air in the lungs, and then that air moves out to equilibrate with the atmosphere. Now, we have pressures throughout this system. We certainly have pressures inside the lungs of the gas. We also have pressures outside the lungs, between the lungs and the wall of the chest cavity. Here's why this is really, really, really important, and I want you to probably write this down. Here's my little animation here of ventilation, if you will. We're changing volume, which changes pressure, which causes equilibration. The lung is not physically attached to the pulmonary chest wall surface. It has to slide. As you're changing volume, there is a shear there. This is true in your heart as well. This is why you have a pericardium around the heart. You also have a pleural membrane around the lungs. It's a fluid filled, because as you're changing volume, there's a little bit of a shear. I'm rubbing my hands together, like my hands are kind of like in a prayer position, and then I start sliding one hand up and one hand down. As you're changing volume, those tissue surfaces are sliding past each other, and so you have to have lubrication. But here's what you don't want. You want that lung to be maximally inflated at every volume. So I want you to look at the animation here on the left. You can kind of see the lung in here. It's kind of inferred in this sort of reddish area. You can see that reddish area never separates from the chest wall. It's not physically attached. There are no ligaments. There's no connective tissue that attaches that lung to that wall. It slides, but it stays maximally inflated whether you're inhaling or exhaling. If this lung separates, we call that a pneumothorax, and it is life-threatening, because if it separates from that chest wall, now you can't change the volume of the lung and you can't move air. So one thing we have is we have a fluid-filled membrane around the outside, and that gives us a little bit of an adhesive sort of a, but yet slippy connection. So like, for instance, if you've ever had a paper towel that's wet, you notice it'll kind of stick to your hand a little bit. There's a little bit of a kind of a fluid adhesion there. The other thing, let me point this out here, is that if we look at the pressure inside the lungs, it would average 760. That's our typical atmospheric pressure, right? It's going to be the same or very, very close to the atmospheric pressure. All right, so we call that intrapulmonary pressure. Now I want you to look at the intrapleural pressure. So if we're at 760 inside the lungs, between the lung and the chest wall, we're at 756. All right. We actually are 4 millimeters mercury. It doesn't sound like very much, but it is a life or death chain difference. What that does is that creates a little bit of suction and pulls the lung up against the chest wall, so that when you're changing volume, it is maximally inflated for whatever space is there. So the difference, we call the transpulmonary pressure, is 4. So it's 4 less than this one. So the absolute value is 4. That's what that means. But it's there in order to keep that lung maximally inflated regardless of volume. Because if it's not, you're not going to be able to change its volume when you inhale and exhale. Very, very important. So extending on the previous slide and bringing back earlier discussions, we're always moving from higher to lower pressure. That's that bulk flow. The pressure in the lungs is proportional or related to the volume of the alveoli. So as we increase the volume, the pressure in the lungs drops, and that causes air to move in. Or if we're exhaling, the pressure of the volume decreases, the pressure rises, and the air moves out. This is actually described in a law of gas physics known as Boyle's law, which says that the pressure of a given amount of gas, so if you have a given number of gas molecules in a volume, if you change that volume, it's an inverse relationship. So if you have a given amount of gas, and what happens is you reduce the volume, the pressure will rise. If you increase the volume, the pressure will drop. So when lungs are expanding, there's a given amount of gas already there. That expansion then with that gas causes a very, very slight, maybe for a hundredth or a couple hundredths of a second, a decrease in pressure, which then equilibrates with the atmosphere. And so this is Boyle's law. The simple concept is that the pressure of a given amount of gas is inversely related to its volume. I think I have a slide on the next slide for you to kind of illustrate this a little more simply than this picture right here. Let me show you what I mean. So this figure comes from another physiology textbook, and I don't want you to worry too much about this left side. I want you to focus on the picture. In this picture, we have a volume that's 1 liter, and there are 1, 2, 3, 4, 5 molecules of gas. And you can see them bouncing off the walls and moving around. That's what actually pressure is. It's the force of the molecules interacting with the surface of whatever container it's in. And so we've got 5 molecules here, 1 liter. Okay, so 5 molecules per liter. If we take that and we reduce the volume by half, so we have half a liter, we still have those 5 molecules there, but the pressure has increased because the concentration has increased. Right? We have 5 molecules in half the space as we had here, so the equivalent density is now 10 molecules per liter. And that's twice as many interactions with the wall of the vessel, which means twice the pressure. So if we define this pressure as 100 millimeters of mercury, that same amount of gas, same number of molecules, when we cut the volume in half, is now 200 millimeters of mercury. It's just that simple. Don't let the math get in your way. It's saying that that with a given amount of gas, if you change the volume, you change the pressure. If we increase volume, the pressure drops. If we decrease volume, the pressure rises. I'm going to take a pause right here. We've gone for about half an hour, which I think is a decent amount of time for a video lecture. So I'm going to pause right here, and we will end part 1 here, and then we will pick up with part 2 with the remaining slides. So I'll talk to you soon. I hope you've enjoyed this lecture. Please let me know if there's anything I can do to help you understand the material better moving forward. Thanks again.